Now we're looking at this figure, which is from page 297 of your textbook. This figure is a representation of how something called the Jeffress coincidence model works. It's described in your book starting on page 296. Now, the Jeffress coincidence model is a very simple neural model to understand how it is we localize sounds coming either from our left or our right side. And basically, the way it works is the theory is that there's a feed or an axon coming from each ear. So there's a feed coming from the left ear and a feed coming from the right ear. And these feeds at some point go into this little circuit here. And this circuit is set up with a bunch of different what are called coincidence detectors. In other words, they will fire only when both inputs are fired. So for example, if this number 4 here, if this one is lit up and this one is lit up, then they will both fire. In digital electronic terms, it's basically like an AND gate, where it'll only fire if this one and this one are stimulated. And essentially what's happening here is that it times how long it takes for the signal to travel from one side to the other. So if you're listening to something that's dead in front of you, right in front of you, the sound is going to reach both of your ears at the same time, which means that the ears, that the neural circuits connected to your hearing are going to be activated at the same time, both on the left side and the right side. Since they're both activated at the same time, the signal will travel exactly the same distance at exactly the same rate. And ultimately, when it goes into this Jeffers circuit, it'll start for the left ear, it'll start here and travel across. So the right ear, it'll start here and travel across. And since they both started at the same time, they'll both end at the same time in the dead center of the Jeffers circuit. Whenever that neuron in the center is lit up, that indicates that the sound is coming from directly in front. Now, if we look at an example where the sound is coming from the right, we see that the sound's coming from the right, so the right ear was stimulated first. So it's got a head start. It starts going into this Jeffers circuit. A few milliseconds later, the left ear stimulation comes in, and it goes in. And ultimately, they both meet up and stimulate the third coincidence detector that's in this circuit. And whenever that detector is lit up, that tells your brain that the sound came from the right side, or from the right side. And that's basically how that circuit works. Um, the figure here. There is some physiological evidence for the fact that we have neurons that respond to different time differences, right? So here we have a situation where we found 6 different neurons that respond only when the sound is coming from a particular place. So, for example, this neuron here only responds when the right ear is stimulated first, and this is in these snow owls. This neuron here only responds when the left ear is stimulated first, and we have actually found neurons that respond very nicely in a line only when at a certain interoral time difference. And this gives us some really good actual physiological evidence for this Jeffers circuit that we showed you before. There are also what we call broadly tuned interaural time difference neurons. The For example, in gerbils, they're in other words, these are neurons that are not very specific, but rather work in a group, work as groups. So in gerbils, we have found neurons in the left hemisphere that respond best to sound from the right hemisphere, and vice versa. Neurons that are in the right hemisphere respond to the left hemisphere. So we do have this separation, right? The separation of sounds that are coming from the left are processed in the right, and sounds that are coming from the right are processed in the left. And these neurons are found in the opposite sides of the brain. The location of the sound is correlated. The way the sound is localized is correlated with the activity of these groups of neurons. So, for example, if the sound is coming from right in front of the gerbil, there will be equal activity on both sides of the brain. If the sound is coming from the right, then there will be more activity in these broadly two neurons on the left side of the brain. So here's an example of these types of neurons, right? So we have in the gerbil and in the owl, we have along the x-axis, what you see is time in microseconds, and then you have firing rate, which is how much the neurons are firing. And what you see here, which is described in your textbook, um, which you see in your textbook on page 298, you see that the main difference between these two curves is how broad the gerbil's curve is. It's a lot wider than the owl's curve. That's because the gerbil brain, it's a mammalian brain. Gerbils are mammals, right? So mammals tend to have more sophisticated cortexes and processing than birds and reptiles do. And as a result, you see broader activity here because there's more neurons involved in processing this interoral time difference as opposed to the owl. The owl only has this very narrow processing, which it's only 80 microseconds, but there's a lot more responding over a longer period of time in the gerbil because there's a lot more of these broadly tuned neurons involved in the gerbil brain as opposed to the owl brain. And the other good thing about this for gerbils is that the neurons, this broad response of neurons, this means you have a lot of neurons that are responding to a lot of different time differences. The normal time differences that are found in nature, in order to be able to detect a difference between where sounds are coming from, is indicated by this shaded area here. So as you can see, the the curve is much broader than the shaded area. And what this means is that this gives the gerbil a much better ability to detect variances in the time differences. Whereas with the owl, really, this is their entire ability right here, which corresponds roughly to this width right here. They don't have as much of an ability to detect different variations in interoral time difference because they don't have these groups of neurons that are working together to look for different time differences across a very broad range of possibilities. This figure is from your book on page 1216, and basically what they did here, or I'm sorry, 12, figure 1215, I'm sorry. And basically what they did here was they recorded neurons on a single trial that were oriented in a spatial pattern to fire whenever sound appears in a certain location. So the sound stimulus in this example came from the 60 degrees, which is right about here, right, 60 degrees away. And basically what they found was that when the sound was coming at this monkey from 60 degrees, that the neurons that were associated in the cortex with this 60-degree area fired the most. And then they fired the least here, of course, on the opposite side. Here's a gerbil, right? This is a little gerbil. And what you can see here are differences in the firing of the neurons in the left hemisphere and the right hemisphere. So, as we said before, if the left ear gets stimulated first, you're going to see the right hemisphere neurons activate first. And then similarly, when the right ear gets stimulated first, you will see the left hemisphere neurons stimulated first. Here in this area, where the interoral time difference is not is very small, is where it's where the animal will perceive that the signal is coming from the front. But once the interoral time difference goes beyond these dashed lines here, that's where the animal can be able to detect differences, and we can see those differences in the way their neurons are responding. Similarly, we see this. This is this figure here. Once again, is showing the firing of neurons whenever the sound is coming from dead on. We have equal activity in both the left and the right. But whenever the sound is coming from the left ear first, the right is stimulated more, and vice versa. If the right ear is stimulated first, the left neurons are stimulated more. Um, these neurons, we have been able to identify these broadly tuned neurons in area A1 and the auditory belt area of the cortex. We have research that from Malhorda and Lomber in 2007, and basically what they did was they did a couple of different studies. One study is where they cooled this part of the brain by putting in little apparatus in that part of the brain to lower the temperature of that part of the brain in monkeys. And when you do, you reduce the firing of that part of the brain. And sure enough, when they do that, they're able to eliminate some of these effects that we just saw with broadly tuned neurons. Um, if you recall before, we talked about different pathways. There are different pathways for auditory information that tell us where things are located. And of course, there are different pathways for auditory information that tell us what things are. The what stream of flow of information is called the ventral stream, which kind of goes from the cortex down. So it starts in the anterior portion of the core and belt and extends to the prefrontal cortex. The what stream, this information that feeds from the anterior portion of the core and belt and to the prefrontal cortex, seems to be responsible for helping us to identify what we're listening to, what sounds are. The other stream is the where stream, and it starts in the posterior core. So instead of the anterior, it starts in the posterior core and belt, and it goes from there to the parietal lobe and the prefrontal cortices up in front of the brain. That helps us to localize where sounds are coming from. So we have these two separate flows of information from the auditory cortex to different parts of the brain, one of which tells us what helps us to identify what we're listening to. The other tells us where it's located. We have a lot of data from brain lesioning studies as well as from looking at human patients with brain damage, like from strokes, that lends a lot of support to these different parts of the brain. So here's a cartoon picture, right, of these areas. So if we look at the core part and this area that we're the A1 area that we're talking about, the where area, uh, which is the dorsal stream, goes to the prefrontal cortex, and then the ventral stream, the what, travels down the bottom. And again, we have been able to detect differences. For example, if you receive a lesion that affects the flow of information in your what stream, you might be able to tell me where a sound is located, but you won't be able to tell me what you're listening to. We can deactivate the what pathway by cutting it here, and that we can do that with a lesion or by cooling that area. and you can't identify sounds. We can also deactivate the wear pathway by cooling or cutting here, and you can't locate where sounds are. This is a picture of two patients, right, JG and ES. JG had brain damage right here, affecting the ventral pathway, and he was completely unable to tell what. He had very poor recognition on what he was listening to. Similarly, we had this other patient here, ES, that had damage to these parts of the brain here. Notice it's affecting the dorsal pathway. This damage right here, and this patient could recognize sounds pretty well, but had a very tough time localizing where those sounds were coming from. One of the interesting bits of research that has a lot of practical value in this area is hearing inside of rooms. Lots of research in this area because obviously it's an important it's important for like architects and for designers of other types of places that want to control the way a room sounds. The direct sound that you hear is the sound that comes from the source to your ears, right? So in a particular room, if one of the things that you're going to hear is the direct sound. The other thing that you're going to hear are indirect sounds, and that's going to be the way the sound reflects off of the walls and then goes into your ear. Every room has a particular profile of the way it reflects sounds. When you're outside, obviously, being outside, you don't hear very many indirect sounds because there's not a lot of places for sound to bounce off of. That's why it's very easy to tell if you're inside of a room or outside. It's extremely easy to tell if you're blindfolded. You can tell if you're inside or outside just from the way the sound is bouncing around or not bouncing around towards your ears.